Today we're going to have a look at a series of experiments on plants that demonstrate how plant growth occurs and how hormones are involved in this process. Now the very first experiment here, we're going to focus on the tip of the plant, and that tip of the plant is called the coleoptile. So first off, let's just consider what would happen in a normal situation or a control. The plant would grow and it would just continue to grow under the right conditions, and up it would go. And what we notice when we get a pair of scientific scissors here and we remove this tip off the plant, the growth stops, completely stops. If we actually are able to return this tip to the plant, keep both ends of the plant healthy and return that tip, what we notice is plant growth will continue. So what that tells us is that tip of the plant, the area right at the end, is involved in plant growth. So scientists having inquiring minds played with this a bit further. And what they did is they did a similar thing. They cut the tip off, but this time they put a little bit of agar in between that shoot tip and the rest of the shoot. And what they noticed here was that when they did this, growth continued. Okay? And that shows us that the message that was coming from the coleoptile wasn't cell-to-cell contact. It was actually something that was diffusing through the plant, from something being released from one end to another. The interesting thing that we found is when this growth occurred, it looked something like this. And the growth occurred underneath the agar. The coleoptile, or the tip, was releasing the message, but the growth was occurring down here. And that's a very different picture than what would occur and what it would look like if the growth had occurred only in the coleoptile and the shoot, the message hadn't been passed to the rest of the shoot. So let's have a look at what might actually be happening in these cells in that growing region of the plant. Now, imagine this is just a microscopic view of some of the cells in that region. Okay, we can see the individual cells and the poorly drawn nuclei. So firstly, what we see is a lot of cell division occurring and the cells just reproducing. So we end up with more cells, which is making the plant basically getting taller. The other thing that we can see that these chemicals are doing is they're creating cell elongation. So the cells are actually becoming a bit longer, okay, which again is increasing growth. So let's recap what we know already. The message being released here at the tip, at the coleoptile, it diffuses, so it's a chemical being passed through, not cell-to-cell contact. It acts in the growing region, we know the cells expand. The probably next logical question for scientists was, what is that chemical? What's happening there? And what they could do is they could actually pull this agar block out and isolate that chemical. And that's exactly what they did. And what they found was a number of different hormones. The one that's probably most common that we're going to be talking about today in these experiments is a chemical called auxin. And that's a very common plant growth hormone. So scientists could then really drop this chemical onto little agar plates on top of the shoots and see if it had any effect. And what they observe is very little growth occurring when there's no coleoptile and no chemical being released. But when they put the auxin on the agar plate, or they take one of these agar plates that they know has auxin in it, they find that there's quite a lot of growth and the plant will continue to grow up. And that's just really reinforcing the evidence that it is the auxin causing the growth. Now I love this next bit because I can imagine working with these little tips of plants and putting little agar bits in and getting it all happening. It would be really fiddly work, and I reckon I'd just be probably a little bit clumsy to get it all spot on. And the next thing that the scientists notice is that sometimes when they put the tip like this, the experiment didn't quite turn out as planned. So let's just have a look at what's happening here by imagining this hormone auxin being released. So the coleoptile is the area that's producing this hormone. I'm just going to draw the hormone as little red dots, and you can imagine that being the chemical. That diffuses down to the plant here. And what we have here is lots of auxin diffusing on one side, but not on the other. When we have that happening, we have lots of growth on one side and very little growth on the other. That causes a bit of a curved growth. You can imagine more growth happening on this side and little growth on this side. A lot more cell division and cell elongation on that side, and that's going to cause a bit of a difference in the growth that's going to occur. To look at that in a cell diagram, really what we have is our cells that we saw before, and we have this side over here, the side that has the more auxin. More cell division, more cell elongation, and less on this side. And we can imagine if this was happening, that we would have, and we could imagine that we'd have a lot more cell growth happening on this side with the plant rather than on this side. And if there's more growth on one side, that's going to lead to a bit of a curving growth. So scientists could do a bit more with this. What they could do is they could drop auxin in the agar on one particular side and observe this curved growth. When we remove the auxin, no growth occurs. When we add auxin, we get lots of curvature happening. So when this curvature occurs, what they can do is they can actually measure the angle of this curvature by measuring this angle in here, and they can measure how much curvature occurs because of the hormone. So they could measure the effect of different concentrations of auxin. The more concentrated the hormone was, the more curve they observed. So they're the experiments that are really behind discovering the mechanism for this growth and the direction of growth in plants. Let's now have a bit of a look at how this applies in nature in the situations that we know. So first off, we know that the plant shoots, the tips, grow towards light, towards the sun. So what this must mean is that chemical that's being released from the coleoptile and going to the growing region of the plant must be on this side of the plant. And when we go looking for auxin, that's where we find the higher concentrations, in this region here. And that's where we're going to get lots of cell elongation, lots of cell division. So as the plant grows, it gradually grows towards the sun or the light. And we call that phototropism, growing a growth response being a tropism, and photo towards the light, a positive phototropism. So this chemical here that we have collects on the dark side of the shoot tip, collects on the dark side. Now let's have a look at another situation. Let's have a look at geotropism. And we know that when we have a seed down here, we know that our growth response in one area of the plant, it's going to go up. In another area of the plant, it's going to grow down. Two different responses. We call this positive geotropism, growing towards the pull of gravity down here, and negative geotropism for the... Now, when we go looking for the auxin in these plants, it isn't exactly what we might expect. You might expect that we find lots of auxin in this area in the shoots, and in a different area in the roots. We actually find it in the same area, and in both roots and shoots, we find it on the downside. We find auxin on the downside. What we find is that it's an opposite effect. In shoots, as we know already when we looked at phototropism, it causes cell elongation and cell growth. In roots, it has an opposite effect. It actually inhibits growth. It slows growth down. So instead of having the effect of making those cells reproduce quicker and take up more space, causing a bend, it slows it down, causing the opposite effect to bend the other way. Now here's a pretty common sort of graph that shows the difference in roots and shoots response to auxin. On this axis here, we're showing growth. So this is growth and this is inhibition below the line. Along here is auxin concentration. So at the high auxin concentrations, we can see that shoots will have stimulation and growth. At the high auxin concentration, roots, however, have inhibition.